Hi guys, welcome to lecture 2 on mutation. In the last lecture we learned about changes in the DNA sequence, specifically point mutations, that is, a single change in the DNA sequence. It could be a point mutation where a letter's changed, or it could be a addition or deletion of a nucleotide which causes a frameshift mutation, both of which have various effects. However, in both of those cases, you're really only affecting possibly one gene. That is, I can make one letter change, which would potentially affect one gene and affect one protein. And that, of course, leads to some common genetic diseases, like the list that was in the previous slide. Things like cystic fibrosis or Huntington's chorea or muscular dystrophy. And in all those diseases, you simply have like one protein that's affected. In the case of cystic fibrosis, it's a chloride ion channel. In the case of muscular dystrophy, it's the dystrophin protein, which connects muscle cells to the fibers that are inside of them. Then we can have another type of mutation, and these are chromosomal mutations. And so these are deletions or the movement or the repetition or the change in direction of segments of chromosomes. And of course, chromosomes have thousands of protein codes on them, thousands of genes. And so a chromosomal mutation will cause what's called a syndrome. Okay, so somebody who has a chromosomal mutation will have a number of different proteins affected, hundreds, thousands, and they have all kinds of issues, not simply the inability to make thin mucus. Anyway, so genetic diseases or disorders are caused by a mutation of one gene affecting one protein, and they're kind of limited. However, chromosomal defects affect portions of chromosomes with large numbers of genes, and they cause syndromes. So we say that chromosomal mutations are examples of large-scale genetic changes, and those changes take place in a couple of consistent patterns. They can have the recombination of chromosomal pieces, or their multiplication or deletions. So when does it happen? Chromosomal mutations generally occur during the prophase 1 of meiosis. Remember that prophase 1 of meiosis was notable because of this phenomenon called tetrad formation. Remember that when I make gametes, I use the process of meiosis to make my sperm or my eggs, depending on whether I'm a male or a woman. And one of the important differences between the cell division in meiosis and the cell division in the process called mitosis is the fact that I literally exchange parts of chromosomes during prophase 1. So during tetrad formation, remember that homologous chromosomes form a synapsis. They connect to one another, and they can undergo recombination, or they can undergo exchange of material. And when that exchange occurs, you can have some common chromosomal mutations occur. So there's 4 types of common chromosomal mutations: inversions, translocations, deletions, duplications, and then a phenomenon called non-disjunction, which leads to polyploidy. That means you got too many or too few chromosomes. And so let's take a look at those in a little detail. Okay, well, one of the first things I asked you guys to look at in the introduction of this unit was a video on Williams syndrome. And Williams syndrome is an example of a deletion mutation. And in the case of chromosome number 7, there's a deletion mutant. And people with Williams syndrome have all kinds of cool attributes and some deficits that make them a little bit different. And so this loss of a segment on chromosome number 7 gives these people exceptional musical IQ, and they're super social and affable and happy. Of course, they also have a tough time with math and some other issues. But that's a perfect example of a deletion mutation. So there's all kinds of changes, and one of the things that's interesting about people with Williams syndrome is that they have some sort of characteristic facial features that makes them all that much more likable. Another possibility is that instead of deleting a segment of chromosomes, is you can actually duplicate. And so I can see here in my little GIF, I've got segments 4 and 5. They get repeated, and that duplication event can lead to a syndrome like DUP15Q. And so DUP15Q occurs when you have a replication of the segments of chromosome number 15. And there's a bunch of different characteristics associated with people that have this chromosomal syndrome. An example of a chromosomal translocation is Alagille syndrome. And in Alagille syndrome, there's a segment of chromosome number 20 that gets taken off and exchanged with a segment of chromosome, I don't know. Anyway, a piece of it gets moved from the place where it belongs to the place where it doesn't belong. And you have this constellation of people with Alagille syndrome. And Alagille syndrome, you have some sort of characteristic facial features. It makes an elongation of the face. Also gives severe itching. Another type of chromosomal mutation is an inversion. And so inversions occur when a segment of a chromosome is flipped 180 degrees. And if you recollect, I alluded to the fact that DNA needs to be read in a specific sequence. And so since I've switched the direction of the chromosome, I can cause some issues in how it's read. And the example for an inversion mutation is Sathry-Shatson syndrome. And so you have these, again, here we go, we get some facial features that are characteristic. One of the most common chromosomal mutations is polyploidy. And in polyploidy, you have an abnormal chromosomal number. Remember that the normal human chromosome number is 46, but sometimes you have a phenomenon called non-disjunction. So remember, when I make sperm or egg, I should have only one copy of each chromosome, right? So I've got 23 pairs of chromosomes, one chromosome, and in each sperm or egg, I should have one chromosome number 1, one chromosome number 2, one chromosome number 3, etc. Well, in some cases, when that sperm or egg is formed, I don't get one chromosome number 1 or one chromosome number 21. I get 2 in my sperm. And when I go to meet the egg, that makes a person with 3 chromosome 21, and that leads to probably the most common chromosomal mutation in humans, trisomy 21 or Down syndrome. And this is an example of polyploidy. Poly means too many chromosomes. And so trisomy 21 causes Down syndrome. And, I simply have got the case where I have a sperm or an egg. I think typically this happens in the egg. So women in their certain age, and it's really it's not a chronological age. It doesn't matter how old you are. It sort of matters how physiologically old you are. So if you're a woman over 30, they typically do something called a triple screen, and they look at the age of your eggs. And so people that have chronologically older eggs, or physiological older eggs, might be more likely to have kids with chromosome 21. Anyway, you might know somebody with chromosome trisomy 21, and you have some deficits, but you also have a lot of really super happy people that are great fun. So we have all kinds of effects of genetic changes, right? Sometimes mutations can have no effect, right? In the case of a point silent mutation. Sometimes they can have effects that are benign, all right? I can change an amino acid, but it doesn't really affect the protein. Maybe it makes it slightly more or less effective. Most genetic changes, of course, are probably not going to have a change because my DNA library is so big that the chances of a change occurring in the middle of a gene are relatively low. However, the next level is that, most mutations are not going to be good for you. And the only mutations that are really important when it comes to humans are the mutations in the sperm or eggs. That is, mutations that can be passed down. And so that's an important thought when we get into evolution next. Our next unit to end up this chapter is on DNA technology, and I'll see you then.